Thank you. So thank you very much for the opportunity to contribute to this event. It is extraordinarily moving hearing all the tributes that are coming through to Eric through both the European event and I'm sure in the others. You know, pay my own tribute and what I'd like to do is kind of reflect on kind of the wider science that kind of is really calling on us to provide our data across, so kind of reaching really beyond our own discipline of palynologists. So that's what I kind of really like to focus on, pretty much using my own kind of personal experiences of that experience of that collaboration. So it's kind of extending some of the work that Jesse's just talked about, and really I must acknowledge the work of Jesse. You know, without my collaborations with Jesse over the last, crikey, how many years, probably 10, 12 years, much of this work wouldn't be possible. And of course, Neil Roberts, former colleague as well. Get back into here. So I just wanted to reflect before I kind of start on the science kind of stuff, the personal recollections of how I have been impacted by Eric over the course of my career. And I was an undergraduate in the mid-90s, and I was one of those cohorts that started by, I was probably the very last of a set of people who would draw a pollen diagram by hand. And as an undergraduate, I was introduced to Tilia, which we've heard a lot about today already. And it really was fundamental in our ability to quickly visualize our data. And there was nothing more exciting than inputting your data into Tilia and then generating that diagram. And it was that moment where you really saw your data and it was there in front of your eyes and you could really start to think about it. So it's so important for us. I think from memory, the first time I really probably met Eric was in 2008 in the IPC at Bonn. And at the time I was a naive and inexperienced researcher. I still feel like a naive and inexperienced researcher much of the time. But the IPC in 2008, I was going through a slightly challenging time, and he was very supportive of me and supportive of what we were working on at that time. And really just then, at that kind of hinge point in my career, I was only really just starting to be involved in these kind of data compilations and the EPD, and the reinvigoration of the EPD led, you know, really driven, I must say, by Thomas, who is chairing us today, and other colleagues who we've heard of. And really, I kind of must reflect and really just re-emphasize the importance of Eric's vision. And he really was a pioneer in the development of open access databases, 30 years ahead of his time, 20 years ahead of his time within this, really. And now we have sort of dozens of papers that appear routinely about the importance of open science and open data and open methods, and has been emphasized already by others who are in a better position to say these kind of things. Eric was there from the very start and kind of driving this process. So we all have a tremendous amount to thank him for there. So what do I mean by hungry science? I want to kind of use this term. Well, we see increasingly big science questions have a strong desire for large amounts of data, large volumes of data, which can be used to address questions. So I have seen, and I'm sure others have seen, a strong desire for data-driven environmental reconstruction. And that desire is coming not only from within our own community, and we've heard some tremendous examples of how we can use the EPD to explore vegetation dynamics, to explore long-term change, driven really by palynologists. But there is this demand from beyond our discipline for this data as well, from beyond our own community of palynologists. And I think it's very important. I think I remember having conversations with Thomas over the years about, you know, the dangers of non-specialists sort of using our pollen data uncritically. And I've seen people present interpolated kind of pollen curves where you have negative values of pollen data. So there is really that importance of specialist palynological knowledge when we kind of use these spatially extensive data sets. So, you know, we are both, you know, ambassadors, curators, and people who can help these wider disciplines work. And frequently when I sort of give talks, I see other people give talks, we talk about sort of three very broad communities of science who have interest in our data. Climate modelers, and we've heard a little bit already from Florence. Nature conservation, we've heard quite a lot about ecological sciences, of course, today. And Jesse started to kind of really put the spotlights onto human impact and the archaeological communities. So I'm not going to give very much detail at all about any of these particular studies, but just to demonstrate this in some way. And I'm not going to rehearse too much of what Florence has already said. She's more than eloquent in being able to say these kind of things. But we understand that vegetation plays a very important role in climate forcing, and Florence has already described the importance that we assess natural processes and human impacts. And she's shown the results from Gustav Strandberg's paper in Climates of the Past, where using modeled anthropogenic change, land cover change. Using anthropogenic land cover change scenarios, we're suggesting, and here Jed Kaplan's model, where we can suggest that temperatures are higher in Eastern Europe and lower in the Mediterranean as a consequence of anthropogenic land cover change. And I think the important thing to emphasize here is that we heard from Rashid earlier about the role of databases, for example, in data model comparisons, for example, the PMIP project. And kind of the early use of the EPD was really around evaluating the results of climate models. This new work that's been done, really driven, I must say, by Marie-Josée Galliard, but facilitated by all Eric's work, is really now allowing us to place paleovegetation data within the climate models themselves. So instead of just being simply a data model comparison approach, our paleovegetation becomes a part of our climate modeling process. We've already heard an awful lot about kind of ecological sciences and vegetation ecology. We've heard from Thomas about the genetics work led by non-palynologists looking at phylogeography, for example, of the oaks. I want to kind of just sort of draw attention to some other aspects of this. So again, coming from the non-vegetation community, sort of question, there are significant questions, and I'm sure Richard will talk about this later. He's talking about mammals. Thinking about the response or even the role of large mammals to changing ecological conditions through the Holocene. So Jesse and I were involved in a collaboration with the Mammal Research Institute from the Polish Academy of Sciences, who came to us because they were really interested in understanding the response of large mammals, in particular looking at bison, auroch, and elk, to changing environmental conditions over the Holocene. So this was a very detailed study, so led by our collaborators. They were looking at radiocarbon dated remains of these animals with stable carbon and nitrogen isotopes to try to reconstruct foraging behavior. And we were asked if we had paleovegetation data which could be used to facilitate this. And I guess the question is, why did they come to us? And in 2015, we published a series of time-stepped 200-year vegetation reconstructions of forest cover across Europe. Now, I'd be more than happy to admit that these were not state-of-the-art at the time. They were what we sometimes describe as quick and dirty methods, but they were precisely what this community were really looking for, something they could use to integrate into their kind of modeling approaches. And again, without wishing to dwell too much on the study, you can read it. And I'm, really, Amelia Hoffman-Kaminska is the person who should talk authoritatively to this work rather than I. Taking this spatio-temporal database of where these mammals were, it was possible to state that before the Neolithic, so it was an important distinction between pre and post-disturbance situations, aurochs were mostly eating grasses and forbs. Bison were grazing in kind of shrub and tree forest steppe habitats. And what was really gratifying is the nitrogen isotope work was confirming what we could see in our pollen-based habitat data. Interestingly, after the Neolithic, bisons really moved into the forests. And what we see in that post-Neolithic situation is that we're seeing these large animals shifting from grazing open spaces to grazing forested spaces. And in spite of the increasing opening of the European landscape, they continue to remain within the forests. So they became forest refugees. So it's really interesting work combining these other disciplinary approaches to look at habitat plasticity in these large animals. And I won't talk about this because time really doesn't allow, but we've done kind of not dissimilar work looking at red deer distributions, and again, led by scientists at the Mammal Institute, really looking to understand habitat change and habitat preferences for red deer. And so kind of being able to show that there are climatic limits to red deer, and they essentially inhabit forests pretty much throughout the late Pleistocene and Holocene. So kind of moving on, and we've had a bit of climate, a bit of ecology, and I just wanted to say something about archaeology as well. And I would classify myself really as an environmental archaeologist if I was kind of into the wall and asked to do so. And Jesse's already talked about some of these collaborations that we've had. But just as within our own community, we recognize the importance of spatially extensive, open, big data. This is a really big sort of driver and move within the archaeological community at the moment. And we're probably all familiar with this pattern of spreading of the Neolithic within Europe, from its origins in the Near East, spreading across Europe and arriving on the far kind of north and west by around 6000 BP. A really interesting, it's a slightly faint slide, a really interesting kind of patterns emerge later as well. So this is a slide of Mette Lovschals from a paper published last year, showing the dating of the establishment of field systems, kind of land tenure boundaries across Western Europe. And already by 4,000 years ago, if we look at Britain, much of that dark grey, people have built large-scale field systems across our landscape, across in Denmark and into southern Scandinavia. So there's a huge desire to understand these processes and human impacts coming from those archaeological communities. And within this, our pollen-based work, our use of large databases, will really continue to underpin our understanding of these human-non-human relationships. So in other words, how people have and did respond to the environment. As an example of that, just a very brief example, work that we have done with a group of geneticists, so who do the genetics work from human skeletal remains. They're able to demonstrate the percentage of ancestry in different parts of Europe based on human skeletal material, and then very interested in understanding the different ancestries, the different cultural groups and their impacts on vegetation. And again, just published last year in the PNAS. And what's really fun actually about working with these other communities is the way that they think about our data. We ensure they use it sort of correctly, but their visualization. So for example, here, the way that they have kind of visualized and looked at the data to look at the difference between Neolithic arrival and the Yamnaya culture's arrivals, to look at and to visualize their data in a different way. The moral, the outcome of the story is that the Yamnayas have a much more significant impact than the Neolithic ancestry groups. And whilst I draw that one to attention, I'm aware of ongoing research at the moment looking at the impact of pandemics using synthesis of pollen data, looking at the Justinian plague. So kind of very hot work being led by Adam Izdebski at the Max Planck Institute. There's new research ongoing looking at correlation and causation of societal development through a big new 10 million euro ERC synergy project led by Christian Christensen. And again, our pollen data that we've produced will continue to make contributions to that. So my kind of final thoughts and final comments really are that there's a clear desire from beyond our own walls, or beyond our own community, for this sort of data that we've been able to will generate. I think it's important that we continue to kind of drive and collaborate within here. And I would say that without Eric's tireless work over the decades and the work of those people he's inspired, really, very little of this would actually have been possible. And our collaborators from other disciplines, Adam Izdebski, Kristen Christensen, Stephen Shannon, all these other people, they won't have known Eric. They may not even have known his name, but they will undoubtedly have benefited from his service to science in its broadest sense. And kind of a final comment, really. Florence showed the reconstructions that we've been doing within the Landclim 2 project. I did a very quick back of the envelope calculation, and I estimated there are 59 million separate pollen identifications that have gone into the Landclim 2 reconstructions at least. And my back of the envelope calculation had that as about 230 years worth of pollen counting. If we just consider someone looking down a microscope, not prepping the slides, not going into the field and collecting the samples, but there's 230 years worth of data just there. And again, without the vision, we wouldn't be able to benefit from those hundreds of years worth of work that collectively are being contributed.